This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is a silencer in regulation of gene expression? Answer: a silencer is a DNA locus where regulatory proteins bind, decreasing expression of a gene on the same chromosome. Question: can you explain what epigenetics is in relation to gene expression? Answer: epigenetics refers to changes made to gene expression without a change in underlying DNA sequence. Question: what are the primary mechanisms of epigenetic change? Answer: the primary mechanisms of epigenetic change include DNA methylation, histone modification, and noncoding RNA. Question: how does DNA methylation affect gene expression? Answer: DNA methylation is the process of adding a methyl group to the DNA molecule. This modification can cause a change in the expression of genes by altering the accessibility of the genetic material to the cellular machinery responsible for transcribing DNA into RNA. Question: can you explain histone modification and its role in gene expression regulation? Answer: histone modification is the process of adding chemical groups such as acetyl, methyl or phosphoryl groups to the histone proteins that DNA is wrapped around. These modifications can affect the structure of chromatin and the accessibility of genetic material to the transcription machinery, leading to changes in gene expression. Question: what role do noncoding RNAs play in the regulation of gene expression? Answer: noncoding RNAs are RNA molecules that do not code for proteins. These molecules play a role in the regulation of gene expression by binding to specific regions of the genome and influencing the activity of genes. One example is microRNAs, which bind to specific sequences in messenger RNAs and repress translation, leading to decreased expression of the target gene. Question: how does silencer work in regulation of gene expression? Answer: silencers are specific DNA sequences that can bind to repressor proteins, which are regulatory proteins that can decrease the expression of a gene. The repressor proteins bind to the silencer and prevent the gene from being transcribed into RNA, thereby reducing the expression of the gene. Question: how is epigenetics different from genetic mutations in relation to gene expression? Answer: genetic mutations refer to changes in the DNA sequence of a gene, while epigenetics refers to changes in the regulation of gene expression that do not involve changes in the underlying DNA sequence. Epigenetic modifications can include DNA methylation, histone modification, and noncoding RNA, while genetic mutations can include substitutions, insertions, or deletions of DNA base pairs. Question: how do epigenetics and genetic mutations interact in regulation of gene expression? Answer: epigenetics and genetic mutations can interact in various ways to regulate gene expression. Genetic mutations can affect the activity of enzymes involved in epigenetic modifications, leading to changes in gene expression. Similarly, epigenetic modifications can affect the activity of enzymes involved in DNA repair and replication, leading to genetic mutations. The interplay between these two mechanisms can ultimately affect the activity of a gene and its expression.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.